Faith and Science program. Be blessed as you watch.
When the seed of a man mixes with the seed of a woman in the body and they make an embryo, a baby, it's very difficult to know what sort of sex that child is going to be. So it's better before you plant that seed into the woman's womb to find out exactly whether it's a boy or a girl. So this is the technique called PGD, Implantation Pre-implantation, that before you plant that seed, this means before you plant. Genetic, something to do about genes. So you want, before you plant, you want to select the gene, either as good gene, or there's boy or girl, or sickle cell, or some other conditions that might affect that baby. Diagnosis. Diagnosis is to find out exactly what is wrong with that. So we can determine the sex of that embryo, or that would be the fetus or the baby or that human being, before actually it's implanted into the mother's womb. I have a picture here about the uterus. This is the uterus. So when the baby is formed from here, see these embryos, they have about 5 to 10 days journey. This embryo cannot be able to tell whether it's a boy or girl. It just goes, it's just random, and implants here in the middle here. But before this goes there, we want to know, if you wanted boys or girls, we want to know as scientists what type of child is going to be. So now we can be able to do it. And for the first time, about, as I said 2 weeks ago, I had a team from Spain and then a team from Brussels. They came to our clinic here in Bokoto, and they were able to do this for us. So imagine this is an embryo. An embryo is a mixture of a seed of a man and a seed of a woman. They join together and they make embryo, something like embryo. This embryo contains all the characteristics of that comes from a man and from a woman. That woman's side, the woman of course has a father and a mother, so all those characteristics that from a man and this is a woman. So each one produces a seed. This is a sperm and this is an egg. The two, they join together and they make an embryo. So all the characteristics from your dad and your mom will be reflected into the embryo. But what we don't know, what sort of sex is this child going to be? We don't know. This embryo is formed. I've got 7 girls. I want a boy. How do I know? I don't want to keep on getting girls because it's very expensive also. And also my wife is getting old. We want to have time to look after our kids. If she produces another girl, then it's another, I'll be disappointed. Also, the family and the community will start wondering, And those who believe that man who doesn't produce boys is not a man, then I will not even stop. I will stop even going to even to the weddings, to attend weddings, because they are going to ask me, how are your boys? I said, I don't have. So what we do, and that's why IVF technology, which is now 34 years old, since it was started in the UK when we had the first IVF baby. It's about 34 years ago. Here in Uganda, it's just been here about 7 years. The first clinic that we started was our clinic here, which started IVF. First time in Uganda, East Africa, and Central Africa. So now this PGD has been in progression almost about 20 years ago. But here for the first time, after maybe 20 years later, we are starting it here. Let me explain to you how this one works. So you have an embryo here. You've done IVF, and you've created so many embryos. Let us say, last week when we did this for the first time, we had a lady who actually had 16 embryos. All these are embryos. Let us, for argument's sake, make 9 embryos. These 9 embryos. So what we do, we don't know what this embryo number, and name them A, B, C, D, E, F, G, H, I, J, this K, L. Okay. So what we do in the lab, we take a small biopsy, as you can see that clip. We take a small biopsy from each one of them. I use the red color. I'll take one of these. I need to get another one. I don't have red color, I will use green. We take a small biopsy, this A, and then we take another biopsy here. We remove this cell, this is B, and we go on doing this. So this is C, and we go on doing this on each of these embryos. These embryos, as I've said, they can progress to become human beings. Not every embryo becomes a human being, because some of them die, some of them are not good, but let us assume they all can progress to become human beings. But I want a particular type of sex, I want a boy. So what we do in the lab was to take each sample of each of these embryos, as I've mentioned. We name them and say A, B, C, D, E, F, G, H, I, J, K, and L. So we took what we did 2 weeks ago. We took a little bit of tissue from each one of these, as you've seen on that clip. We brought one doctor from Barcelona, Brussels, who has done this for us, and he's taken each sample. The following day, she flew back to Barcelona and went. Within 8 hours, we were able to know that this A, example, this A is a boy, B is a boy, C is a boy, and all these, 5 of them were girls. And also we got some good information. We found 1 of 2 were Down syndrome. this embryo, these two embryo, J, these two were Down syndrome. I'm going to show you a picture of a Down syndrome. Down syndrome baby is babies who look like Chinese. Their face is like Chinese. Their tongue comes out, and they're mentally subnormal. They tend to have heart diseases and so on. Most of them don't even survive for after the age of 20. And usually when you have these kids, the parents, they tend to suffer quite a lot and so on. So we're able to also to discover there were 1 or 2 baby embryos were Down syndrome, and the others actually were also abnormal. So this process of PGD also tells us a lot of things about embryos, whether they are normal or abnormal. And also, in cases of sickle cell, we can also be able to know this child, this embryo is sickle cell free, or this is affected, and so on. So this is the beauty about this technique called pre-implantation genetic diagnosis, PGD. So in this case, we found this couple. Out of the embryos that we did, let us say there were 9. They have only 3 boys, and the rest are there are 4 girls, and the others were abnormal babies. So if we had put these embryos, supposing we had selected at random and replaced back these 4 embryos, we may find actually they are girls, and then that man will say, well, doctor, I wanted a boy, and then because I don't know, I don't have any way of knowing. But by taking a small tissue out of each biopsy, for example, you can take, without seeing my face, somebody can come and cut a small skin, take a biopsy from my skin here, and you can go in the lab and tell that this person is a man, because you can tell whether there's X and Y. You can also tell whether I'm white or black by looking at my skin. So in the same way, we're able to do this technique for the first time ever in our country and East and Central Africa. So we are waiting for the results to see if that lady, they will become pregnant, and of course they will get what they wanted. It's time consuming, and of course it's very expensive, but there are people who are ready to prepare for it, and we'll try as time goes on to make it much cheaper and cheaper, so that we can give families, balanced families, and everybody will be happy. And I'm just going to introduce this lady, Elaine from Barcelona. She's a young doctor, she's a genetist, looking only on genes, and she's going to explain a little bit more in technical part. And then next week we are going to also to see other conditions that can be helped. There are about 1,500 medical conditions that we can be able to help, so that you can have a healthy child. There are babies called Down syndrome, as I mentioned, is the commonest anomaly, which tends to affect women who have children after the age of 40. So it's more common in that group. So if we can be able to exclude, especially if people have had Down syndrome children, before they plan for another child, they should go through this system so we can choose a right baby to put back. This technique now is here. We thank God, and we thank the government to support us in many ways, so that we can be able to carry out our scientific advance. Before, as I mentioned, this lady is explaining to you, before I end this program, I wouldn't like to end without inviting you to know Jesus. Because Jesus is the reason why we're here. I'm not here to tell you about religion or anything else. I just want to talk about Jesus. I'm not talking about Church of Uganda, Seventh-day Adventist, Catholics, Muslims. We are talking about Jesus. And Jesus made a challenge. He made an invitation. He said, what shall it profit a man if you gain the whole world? If you gain the whole world. If I owned Uganda and everything that is in it, supposing I owned Kampala and everything, all the houses, everything. But the Bible, Jesus said, what is going to benefit you, profit you, if you are to be the richest man in the whole world, but then you lose your own soul? Father, Lord Jesus, we thank you for this program. Thank you for my listeners and my viewers. May you bless them. And thank you for those who have accepted you, that one day they will be with you. In Jesus' mighty name.
We work with an stereo microscope. I carry this from Spain, and then we use hypotonic solution. This is a solution who makes the cell bigger. Then it's more easy for me to take the cell, the embryonic cell. This is a system to work with and to take the cell from the dish and to put the cell on the slide. And this solution is made of 3 parts of methanol and 1 part of acid acetic, and it's called Carnoy. And this solution works destroying the cell but fixing the nucleus on the slide. This is what we want to have, the nucleus. And this is a kind of pencil who can write on the glass. So I write the number of the embryo on the glass to record the number. And when I will arrive to Spain, I will put proof on this slide, and later we will analyze the nucleus, and we will see if there are boys or girls, and we will call Dr. Sally and tell him which embryos are boys and which embryos are girls. We start our collaboration with Dr. Sally. It's our first time in Uganda, and we are going to do 2 types of cases. First, we are going to do sex selection, because there are a lot of families that they want to have a boy. So we are going to do these cases, and we also are going to do to analyze sickle cell anemia on the embryos, because there are a lot of patients who have this illness, and they want to avoid that the children have also this illness. So basically we are going to work in these 2 kind of cases, but we can work also with other single gene disease or other chromosomical abnormalities we can do. And I think we are going to come two times to Uganda per year. One now in January and another one in August, during August, the last week of August. And we are going to do cases in collaboration with Dr. Sally and also with a team from Belgium. They are doing embryo transfer now. So if you have any questions, just ask Dr. Sally, and we are very happy to help you. This is a human embryo on day 3 of development. It has between 8 and 12 cells, and what we need is to take only 1 cell of the embryo to analyze them. What we do is first we make a hole on the zona pellucida, that is this part of the embryo who protect the cells. We make a hole with thyroides acid, like this, and then we just take one of the cells with a pipette. This is 1 cell. This cell has the same genetic information as all the other cells of the embryo. we take this cell and then we fix as I showed you previously. Then when I arrive to the lab, what we do is to put some proofs that have different colors. This kind, and this is a nucleus, a human nucleus from a cell, from an embryo, and these different proofs are in different colors of the embryo. In this case, we have 2 signals for the 13 chromosome, 2 signals for the 16 chromosome, 2 signals in blue for the 18, 2 signals for the 22, but we have 3 green signals that it means that this embryo has a 31 trisomy and he has a Down syndrome. So we discard this embryo. This is another kind of analysis. It's called PCR. This kind of analysis is performed for sickle cell anemia. And what we found is we amplify different parts of the genome and we analyze. And depending of the peaks that we found, we can tell if the embryo is ill or if the embryo is healthy. This is the path that we analyze. What we do first for PGD analysis is to place 4 drops of PGD medium in this glass. Under the stereo microscope, we place 4 droplets of media and we place 1 embryo in each drop. Then we take the dish with the embryos and we go to the inverted microscope. It has the objectives in this side. So we do with the pipette, we hold the embryo with this pipette and we manipulate with this to hold the embryo. And with this we can move the embryo if we want to take the cell. Here. What we do is to hold the embryo with this pipette, and then here we have 2 pipettes, one with the acid and one for the biopsy. We make the hole with the one with the acid and then we change pipette to do the biopsy. Once the biopsy is done, we make all the 4 biopsies at the same time. Then we take the dish back to the current flow. We just take out the embryos. We leave the blastomeres here, and with the embryos we go to the incubator here. We leave them here, just growing until day 5.
Thank you so much for watching Faith and Science. Your calls and SMS have made a very big difference. At Faith and Science, we are about revealing God's wonders. Remember, we are fearfully and wonderfully made in His own image. Come, let us study the Word of God together at Women Hospital International. We love you so much. May the Lord always bless you.